Bismillahirrahmanirrahim. Dear student, the next topic is from second year of biology, gene cloning. Inshallah, we shall discuss this topic in detail. What is the basic concept of gene cloning? What are the main methods of gene cloning? First of all, we are going to explain gene cloning. What is the meaning of gene cloning? The production of exact copies of a gene in lab is called gene cloning. It is also called to make a photocopy of a particular gene. To make a photocopy of a particular gene. Gene cloning, is the asexual process. Point to be noted. It is the asexual process in which gene copies are produced. Gene cloning is completed in vitro or in vivo. In vitro means that gene cloning which is totally taking place in laboratory outside of the organism body. वो मेथड, वो तरीक़ेकार जिसमें जीन की कॉपी क्रिएट की जाती है और लेबॉरेटरी में ऑर्गेनिज्म के बग़ैर उसके जिस्म से बाहर, तो इस प्रोसेस को कहा जाता है इन विट्रो. What is in vivo? In vivo mean inside the organism body. वो अमल, वो प्रोसेस जिसमें हम जीन की कॉपी क्रिएट करते हैं, बनाते हैं ऑर्गेनिज्म की बॉडी के अंदर. फॉर एग्जांपल, वी क्रिएट अ कॉपी ऑफ जीन इनसाइड द बैक्टीरियल सेल. देन इट इज़ कॉल्ड इन विवो प्रोसेस. Methods of gene cloning. There are two main methods. There are two main methods for the production of a particular gene. The first one is RBT. It is not the Peshawar RBT. It is the RBT. It is called recombinant. It is called recombinant biotechnology. It is also called recombinant DNA technology. There are two main methods for the production of a desired gene. The first one is RBT, recombinant biotechnology method. It is also called recombinant DNA technology. And the second one is PCR. PCR stands for polymerase chain reaction. This RBT method is applied on an industrial scale. When a large segment of gene is copied and produce a lot of product on a huge amount on industrial scale, then the process of RBT is used. PCR is mostly used in forensic science. This method is commonly used in forensic science. What is forensic science? The scientific study and investigation of criminals is called forensic science. In this PCR method, a small segment of a DNA is amplified. In this method, a small segment of DNA is amplified and then can studied for the desired quantities or targets. First of all, we are going to explain the RBT, recombinant DNA technology or recombinant biotechnology. Recombinant DNA technology. It is the method of gene cloning. It is employed on industrial scale. It is a in vivo method. In this method, genes are copied. What are the requirements of RBT? Following are the main requirement of RBT. The first one is gene of interest. It is also called gene of good gene or gene of choice. That particular gene which can be obtained and their copy is produced for a desired character is called gene of interest, good gene or gene of choice. How gene of interest is isolated? Dear student, now we are explain how gene of interest is obtained. How gene of interest is obtained. How gene of choice is obtained. वो कौन से तरीक़े हैं जिसकी मदद से हम जीन ऑफ इंटरेस्ट या खुद जीन या जीन ऑफ चॉइस को हासिल कर सकते हैं? तो इसके लिए 3 मेथड हैं. There are 3 methods. The first one is from chromosome. एक मेथड ये है कि हम enzymatically, for example, it is a chromosomes or DNA. When we apply particular enzyme which are called restriction endonucleases. When we apply the restriction enzyme on this particular chromosome or DNA, the gene of choice is obtained. It is the most common method which is used in laboratories. The second one is artificial synthesis. Scientists can make gene of interest in lab by using a particular nucleotides and enzyme. This method is used when we make a very small segment of a gene. It is a very expensive method. We know that Human Genome Project already sequence human genomes. By using these projects, scientists make the artificial segment of a particular DNA or gene inside the laboratory by using particular nucleotides and respective enzyme. It is the second method of gene of interest. The third method of gene of interest is from mRNA. From mRNA we also obtain gene of interest. How we can obtain gene of interest from mRNA? When we apply reverse transcriptase enzyme, जब हम inside the laboratory messenger RNA पर particular reverse transcriptase enzyme apply करती हैं, जो कि mostly HIV virus में these enzymes are present. These are the special enzymes which have the ability to make a copy of DNA from RNA. These enzymes have the ability. These have the ability to make a copy of DNA from mRNA. This is the messenger RNA. When we apply the reverse transcriptase enzyme in this messenger RNA, it make a copy of DNA which is called cDNA. cDNA means copy DNA, and from this DNA we can obtain gene of interest with the help of restriction endonucleases. Dear student, recombinant DNA technology, it is used for gene cloning. It is used for industrial scale. It is the in vivo method. A requirement of RBT or gene of interest good. It is also called good gene or gene of choice. How gene of interest is obtained? There are three common methods by which we can obtain gene of interest. The first one is from chromosome. When we apply a restriction endonuclease on a particular segment of chromosome or DNA, these enzymes have the ability to cut the DNA or the flanking ends and make a copy of a gene. Artificially synthesis. This method is used inside the laboratory, in which scientists combine nucleotides under a particular protocol and make a proper gene. From messenger RNA, when we apply reverse transcriptase enzyme on a particular messenger RNA, these enzymes have the ability to make a copy of DNA which is called cDNA. This DNA is further analyzed and we can obtain the gene of interest. Student, gene cloning का मतलब क्या है? The production of exact copy of a gene. जब हम जीन की कॉपी तैयार करते हैं, इस अमल को जीन क्लोनिंग कहते हैं. It is also called a photocopy of a particular gene. उन केस में हम एक जीन की फ़ोटोकॉपी की नकल बनाते हैं, इसलिए इसको जीन क्लोनिंग कहा जाता है, या मेक अ photocopy of a gene. Gene cloning is the asexual process. It is carried out by asexual reproduction. There is no involvement of male and female organ or sex cell. In vitro, that process which is taking place outside the organism body in laboratory is called in vitro process. In vivo process, that process which is taking place in organism body is called in vivo process. Method of gene cloning. There are two common methods of gene cloning: RBT, recombinant DNA technology, and PCR, polymerase chain reaction. Now we are going to explain the molecular scissors. Molecular scissors, these are also called restriction enzymes. These are the special enzymes, special set of enzymes. These enzymes are present in bacterial cell. Bacterial cell use these enzymes against viruses. Therefore, these are called restriction enzymes because they restrict, they minimize, they control the activities of viruses. Therefore, these are called restriction enzymes. Bacterial cells have the ability which protect itself from these enzymes. The special methods are adopted. These bacteria which protect itself from the restriction action of these enzymes, and these enzymes are used against viruses. There are 400 restriction enzymes. These enzymes are very particular in action. Some enzyme make a cut at 3' end. Some enzyme make a cut at 5' end of DNA. There are a lot of enzymes. These enzymes target a special sequence. These enzymes target a special sequence on DNA strand, which is called palindromic sequences. Point to be noted. Palindromic sequences. What is the meaning of palindromic sequences? Now we are going to explain the palindromic sequences. Identical sequences are bases. Identical sequences are bases which run. Identical sequences are bases which runs in opposite direction. On opposite direction, which run on opposite direction of the complementary strand of the complementary strand are called palindromic sequences. For example, AATC. For example, AATCG. AATCG. For example, AAGCT. It is a sequence. It is sequence. It runs from in this direction. When this sequence runs in opposite direction, then it is called palindromic sequences. The opposite sequences of these bases are AA, G, C, and T. This sequence runs in opposite direction. Then these sequences are called palindromic sequences. DNA स्टैंड में वो सीक्वेंसेस जो ख़ास तरतीब से या तो 5 प्राइम एंड से 3 प्राइम एंड तक जाती हैं या 3 प्राइम एंड से 5 प्राइम एंड तक जाती हैं. फॉर एग्जांपल, इट इज़ अ सीक्वेंसेस adenine, guanine, cytosine, and thymine. When this sequence become opposite, when we rule this sequence, then it make palindromic sequences. When we fold these sequences, then it make TTAA, CG, GC, and AT. These sequences are called palindromic sequences which runs in a opposite direction. There are a lot of restriction enzyme. The most common is EcoR1. EcoR1 Escherichia coli restriction enzyme 1, BOMH1, Bacillus amphiloquescens, HEN3, and so on. Now we explain 3 overhung and 5 overhung. 3 overhung, 5 overhung, and blunt ends. These are the sticky ends. What is the meaning of sticky ends? Those ends which have the ability to overlap with a particular DNA strand are called sticky end, and those ends which have no ability to overlap with a particular strand are called blunt end. For example, it is a sequence this sequence, it is a hook which can overlap with another strand, then it is called sticky end. And this strand it has no hook, it has it is no hook, it has no attachment site. ये जो DNA स्टैंड में इसमें जो अटैचमेंट साइट है वो नहीं अवेलेबल है for the restriction enzyme, then it is called blunt end, and these ends which have a hook like structure are called sticky end. What is the meaning of three overhangs? When a sticky end is produced it from a 3' end, it is called three overhang, and when a sticky end when the sticky end is produced it 5' end, and it is called 5 overhang, point to be noted. This is called 3 overhang. When a sticky end is present at 3' end, then it is called 3 overhang, and when the sticky end is present at 5' end, then it is called 5 overhang. For MCQs, you note that bomb H1 always cut between bomb H1, it always make a cut between guanine, point to be noted. Similarly, KPN1 it make a cut between CC, it make a cut between CC. SMA it make a cut between CG. These are the restriction enzyme. Bomb H1 always it has a spatial restriction site which is present between guanine. KPN1 always cut between cytosine. SMA it is a particular enzyme, it make a cut it CG sequences. So it is enough for today. Inshallah, in next lecture we discuss about vectors and our expression system. These are the two main requirements of the recombinant DNA technology. The first one is gene of interest. It is also called gene of choice. Gene of interest and gene of choice. Such a good gene is obtained by 3 methods we have discussed it. And these are the next requirement is molecular scissor. These are also called restriction enzyme. These are the spatial setups enzyme. These enzymes are present inside the bacterial cell. Bacteria use these spatial enzyme against virus attack. There are at least 400 restriction enzymes are discovered. These restriction enzyme cut at a particular site. These particular sites are spatial sequences which are called palindromic sequences. Palindromic sequences, these are the identical sequences of bases which runs in opposite direction of the complementary strand. ये वो स्पेशल सीक्वेंसेस हैं जो कि खास डायरेक्शन में अपोजिट सीक्वेंस में ये जाते हैं. इन पर रेस्ट्रिक्शन एंजाइम पार्टिकुलर इन पर पे कट लगाते हैं. इनके जो रेस्ट्रिक्शन साइट है वो 3' इन पे अवेलेबल है या 5' इन पे अवेलेबल है. अगर रेस्ट्रिक्शन साइट 3' इन पर अवेलेबल है और उस पर स्टिकी इन आ जाता है तो उसको कहा जाता है 3 overhang. और अगर restriction site is present it and create sticky in 5 in, then it is called 5 pore hung. और जो ends जिसमें रिस्ट्रिक्शन साइट इक्वल कट लगाती हैं, जिसमें नो हुक या अटैचमेंट साइट नहीं होती, तो उसको कहा जाता है ब्लंट एंड. बाम H1 हमेशा गवानीन और गवानीन के दरमियान कट लगाते हैं. KPN1 जो रिस्ट्रिक्शन एंजाइम है वो साइटोसिन के दरमियान में कट लगाते हैं, उसको काटते हैं. SMA जो रिस्ट्रिक्शन एंजाइम है वो हमेशा साइटोसिन और गवानीन में कट लगाते हैं. इट इज़ इनफ फॉर टुडे.